Hello everyone, welcome to VLearn and welcome back to the class Molecular Basis of Inheritances. In the last class of Molecular Basis of Inheritances, we basically studied about the material which act as the genetic material. And basically these experiments were to prove that DNA act as genetic material. So it all started with the Griffith experiment. Then we studied about Avery McLeod-McCarthy experiment. I encourage you to watch this video if you haven't watched the earlier video. So Avery McLeod-McCarthy experiment. So Avery McLeod-McCarthy, Griffith just said that there was transformation, and he said that something was responsible for transformation. And Avery McLeod-McCarthy gave a strong evidence that this something was basically DNA. So basically they proved that DNA acted genetic material. So this is the further experiment which basically proved that DNA is that something which caused this transformation, the something which caused the change from the R form to the S form is basically the DNA. So Halcyon and Halcyon, another experiment which proves that DNA act as genetic material is Hercy and Chase experiment. So this is the Hercy and Chase experiment. This experiment is also known as equivocal proof. Further proves that DNA act as genetic material. So it is also known as equivocal proof that DNA acts as genetic material. So in the NEET questions, you may find that which of the following experiment best prove that DNA act as genetic material, and you have given both of these three options. That first option is Griffith. Of course, the Griffith experiment doesn't prove that DNA acts as genetic material. Avery McLeod-McCarthy, yes, it proves that DNA is a genetic material, but equivocal proof, the strong proof is Halcyon and Chase experiment. So this is the equivocal proof that DNA acts as genetic material. So it was very recently, it was until in 1952. So it was in 1952, in the year 1952, that Halcyon and Chase experiment proved that DNA acts as genetic material. So basically, he started experimenting with bacteriophage. So Halcyon and Chase, both of these scientists started experiment with bacteriophage. They took the two batch cultures. They took the two batch cultures of the bacteriophage, batch 1 and batch 2. In the batch 1, the bacteriophages are the virus particle which eats bacteria or which infects bacteria. So bacteriophages are basically the virus particles which infects bacteria, such as E. coli cell infects E. coli or infects bacterial cell. So basically he radio labeled the S35 radio labeled protein part with S35 because sulfur is unique in protein. Sulfur is unique. If we talk about the major molecule of a protein and DNA, it's carbon, hydrogen, oxygen, and nitrogen. And these are similar in both bacterial protein and bacterial DNA. So basically they radio labeled the different part, that is S35, because there are some amino acid which contain sulfur. So sulfur is unique for protein. So they radio labeled protein part with S35. In the second batch, In the second batch, the bacteriophage was of course radio-labeled, and its DNA was radio-labeled. And its DNA was basically radio-labeled with phosphate 32 radio-labeled DNA. So its DNA was basically radio-labeled with phosphate 32. So in the first batch, S35 radio-labeled protein was present. In the second batch, phosphate 32 radio-labeled DNA was present. And both of these infected the E. coli cells. So in the next step, they infected. So the first step of this bacteriophage was to infect E. coli. So let's start with the experimental part now. So this is the simple representation for the Hercy and Chase experiment. So we have radio-labeled with radio-labeled sulfur protein. So the sulfur was basically radio-labeled with S35. So S35 radio-labeled protein. So in the first part, this bacteriophage here is with S35 radio-labeled protein. These bacteriophages now infects the E. coli cells. So these cells here are basically the E. coli cells. So the E. coli cells transfer its genetic. Now we know that it transfers its genetic material into the E. coli cell. So let's say this is the genetic material for E. coli cell, and this is the genetic material which is transferred from bacteriophage cell. So this genetic material is transferred from bacteriophage cell to the E. coli cell. So this particular genetic material here is bacteriophage DNA. Because in this particular case, sulfur was radio-labeled. And protein DNA was not radio-labeled. In this next step, so this step is basically what we call infection. In the next step, the next step was blending. In the blending step, they were the bacteriophage which attaches themselves to the E. coli cells. They were rotated with a higher speed, so the blending occurs, and blending means detachment of the bacteriophage particle, from these E. coli cells. So these E. coli cell contain the bacteriophage DNA now, and they do not contain the protein part. And in the third step, there was centrifugation. So if we know the principle of centrifugation is basically based on the principle that the high density particle will be settled at the end, and this part is with the higher molecular weight, and because of the higher molecular weight or the higher density, this form a pellet here. So this is the pellet part, and the upper part is present in the form of liquid, and this liquid is supernatant. So in this particular experiment, if we talk about supernatant is the part with the light density, and pellet is the part with higher density. So if we talk about the higher density, the higher density will be for E. coli cell, and these are of course with less density. So supernatant basically represent the bacteriophage part now. Bacteriophage part without DNA, and pellet part basically represent the E. coli cell. And they measured the activity in both pellet and supernatant, and they find out that in the bacteriophage part, that means in the supernatant part, radioactivity was present, because in this particular case the S35 labeled. So the bacteriophage was S35 labeled, so this shows radioactivity. So radioactivity was present in this particular case. But in the pellet part, because pellet part took the DNA from the bacteriophage, which was basically not radio labeled. So this pellet part do not show radioactivity. There were no signs of radioactivity. That means the genetic material, that's something which is passed from the bacteriophage to the E. coli cell, that something is not DNA, is not protein, sorry, it's DNA. To confirm this, they took the second experiment and did the same experiment with its DNA labeled. So it's radio labeled. They conducted the same experiment radio labeled with radio labeled phosphate 32. The first step was infection. So then now it's phosphate 32 radio labeled DNA. Of course, this DNA gets transferred from the bacteriophage to the E. coli cells. So this bacteriophage DNA is basically now in the E. coli cell. And this is its radio labeled phosphate 32 radio labeled DNA. In the second step, there was blending. Blending means that detachment of the bacteriophage from the E. coli cell. And in the third step, there was centrifugation. Similar to the first case, the centrifuge tube contains the two parts, the pellet part and the supernatant part. The pellet part and the supernatant part. Now, because the pellet represents the E. coli cell, the bacterial cell, and sulfur, supernatant contain the bacteriophage cell. In the bacteriophage part, that is the supernatant part, there was no radioactivity. But radioactivity was present in the case of pellet. So in the pellet, there was radioactivity recorded. That means this genetic material which was present in the bacteriophage cell was transferred from the bacteriophage to the E. coli cells. So, pellet में radioactivity present होना, that means radioactivity present होना in the pellet cells, that means that DNA का जो transfer हुआ है, the DNA is transferred from the bacteriophage cells to the E. coli cells. That means that something which causes the transformation is basically the DNA. और कोई चीज ट्रांसफर हो ही नहीं सकता. Protein, we had checked with protein, we had checked with DNA, and there is nothing else in a bacteriophage cell. Therefore, the DNA is the only material which gets transferred from the bacteriophage cell to the E. coli cell during the reproduction, and this mode of reproduction in viral cell or bacteriophages is basically called as transduction. So this is the equivocal proof that DNA is the genetic material. I hope you liked the video. If you have any doubt about this experiment, let me know in the comment section. Thanks for watching the video, and please don't forget to hit like and subscribe button. Thank you. See you soon in the next video.